Hello everyone, I'm Ozge Gizlenci, second year PhD student at the Abraham Institute of University of Cambridge. In my poster, I presented about a workflow for the transcript characterization of germinal center B cells to reveal molecular mechanism of positive selection. Germinal centers are specialized areas in secondary lymphoid tissues in which B cells undergo affinity maturation. Abnormal functioning of them might lead to malignant disease such as Burkitt cell lymphoma. Germinal centers are composed of two compartments as dark zone and light zone, in which they are positively selected, and transcription factors such as c-Myc is transiently expressed in these positively selected B cells, and it's necessary for the continuation of cell cycle reentry during the selection process to produce high affinity antibodies. As well as transcription factors, RNA binding proteins were observed to have important roles in the selection process. Recently, our group revealed that PTBP1 regulates c-Myc dependent genes, and they're involved in post-transcription processing such as alternative splicing, RNA abundance, and alternative polyadenylation. RNA sequencing is used to obtain qualitative and quantitative measurements of transcript and isoforms. Introduction of long-read sequencing technology helped to solve the problem of exon connectivity in complex isoforms such as OPA1, which has 8 annotated isoforms with the inclusion and exclusion of exon 5 and exon 7. As it can be observed, short reads fail to distinguish any of these isoforms, while long reads can detect each isoform with one complete read. Our workflow for long-read sequencing with Nanopore consists of 4 major steps. During the optimization process of each step, we realized that long-read sequencing with standard Nanopore method is inefficient to use with rare cells due to the requirement of high input RNA, low output of raw reads, low quality of the reads, which can be an obstacle for the reliable data analysis for the isoform detection. In order to improve the output and the data quality, we revised our workflow and went back to the library preparation step and decided to improve our library preparation. We decided to adopt a new library preparation process using SmartSeq2 method, which is a library preparation method developed for high throughput transcriptome analysis of single cells. It's different from standard Nanopore library application. We used 1 nanogram total RNA and a template switching oligoprimer with ribonuclease bases to increase the efficiency of length strand selection, and then we used end-repair detailing in order to have an adapter ligation mediated by enzymes such as T4 DNA ligase. When we compare the efficiency of standard Nanopore method with new SmartSeq2 adapted library preparation method in the sequencing run, we were able to see an increase in the output using a lower input RNA. Our output increased from 1.5 million reads to 15 million reads, showing 10 times increase in the output. In addition, we were able to see a less adapter contamination in the pore occupancy, as well as significantly higher sequencing pore ratio in the pore occupancy. When we observed the regulation of our gene of interest PKM by the RNA binding protein PTBP1 using Nanopore raw reads, there was an increase in the usage of exon 9 in the absence of PTBP1. This was shown previously using Illumina reads quantified by Armat's inclusion analysis. Nanopore was able to recapitulate the conclusion of PTBP1 suppresses the inclusion of exon 9 and the expression of PKM1 in the follicular B cells. Next, we use FLAIR pipeline to observe differential isoform use in the control and PTBP1 knockout follicular B cells. Using 10 supported reads for each detected isoform, FLAIR was not able to detect PKM1 isoform use in the PTBP1 knockout samples, although we were able to observe a 4-fold increase in the use of PKM1 isoform with raw reads of Nanopore. This showed the weaknesses in the ability of FLAIR for accurate detection and quantification of the isoforms for the further downstream analysis. We decided to improve the splice variant detection by developing an additional method For this, we developed an analysis workflow called Nexons with the help of Simon Andrews. After a standard pre-processing of nanopore data, we used BEM files coming from Minimap2, which is used for the detection of highly expressed genes. Then we mapped the reads coming from these genes to genomic segments in order to detect the cDNA sequences and extract splice junction coordinates, which then filtered in order to have the quantitative count table, which is then used for the downstream analysis. Next, we checked our Nexon pipeline to see if we are able to detect PKM isoforms. After counting the isoforms of each highly expressed genes, we were able to see 2 highly expressed isoforms of PKM were detected. Nexon was able to detect the increase in the PKM1 isoform from 3.2% in control to 16.6% in the absence of PTPP1, which is the similar proportion observed in nanopore reads and Illumina RNA results. Our work summarized the challenges of transcriptome characterization in rare cell populations using long-read sequencing. We were able to identify complex isoforms that couldn't be detected with short-read sequencing. Benchmarking of existing computational pipelines for the isoform detection showed the requirement for additional tools for inaccurate detection and for the differential analysis of splicing isoforms. Thank you very much for your interest, and I would like to thank to my supervisor Martin Turner and to our lab, also to Simon Andrews for his help in the development of the analysis workflow.